children, now we are going to start with the next very important topic, and which we have done in +1 also, but I know many of the students are still confused. After doing the process of the microsporogenesis and megasporogenesis, we have done the pollen pistil interaction. We have also studied about the artificial hybridization. Now the pollen tube has germinated and now it has to reach to the ovule. So what we have to do in this section is the double fertilization. Very important topic, double fertilization. The double fertilization was discovered by Navaschin. There was a scientist called Navaschin. He discovered this, and in 1898. This was first time done in Lilium and Fritillaria. Now we will discuss what double fertilization is. We will just go step by step so the things are very clear to you. Now this was the stigma and this is the ovary, and inside we have the ovules. We have ovules. These are the ovules present here. Ovary and the ovules are there. Is that very clear? Now what happens is this, we have done that pollen grain comes from a germ pore. The pollen tube enters by undergoing the pollen pistil interaction, and suppose here we have a micropyle. The pollen tube will enter through here. So let me draw it. In the magnified view, we have an ovule. Inside the ovule, the embryo sac was developed. This was the embryo sac. Let us take this as a micropyle end and this as the chalazal end. At the chalazal end, we had 3 antipodals, and at the micropolar end, we had 2 synergids and we had 1 egg cell. This synergids have the filiform apparatus and we have 2 polar nuclei. Now I told you the pollen tube always enters through the micropylar end and it enters through one of the synergids. We have discussed it already. The filiform apparatus helps and guides the pollen tube to enter here. Now we know that in the pollen tube we had 2 male gametes and the tube nucleus was guiding it. Immediately when the pollen tube enters into this one of the synergid, it gets burst. It gets burst and the 2 male gametes come inside. Now let us discuss what happens the next. This is the embryo sac. Again I am drawing it. These are the synergids. This is a egg cell. This whole is called as egg apparatus. We have 3 antipodals here and 2 polar nuclei. These all are haploid structures. These all are haploid structures. This is also N. This is also N. This is also N. Now what happens is this has got burst and the 2 male gametes are thrown inside. Now what happens in the very first step? One of the male nucleus, we have got how many male gametes? 2 male gametes, right? 1 male gamete, out of these 1 male gamete, 1 male gamete combines with this. What is this? This is egg cell. Combines with the egg cell to form zygote. And this process is called as syngamy. What is happening? Syngamy. That means two gametes are uniting. Got it? This forms a zygote. The first thing we have got is zygote. The second male gamete. The one male gamete come here and unite with the egg cell. Another male gamete goes and unite with the polar nuclei. Now, second step, what happens? The second male gamete unites with two polar nuclei. Now we know each polar nuclei is N, and the male gamete is also N. Therefore, what will happen? This N plus N plus N becomes 3N. They all get united. As the three gametes, or we can say three nucleus, not three gametes, but three nucleus are uniting. That is why this process is called as triple fusion. Why triple fusion? Because three nucleus, one of the male gamete, one polar nuclei and the second polar nuclei. These all three unite to form 3N structure, which is called as primary endosperm nucleus. In the short form, it is also called as PEN. That means after triple fusion, after the development of the PEN, we will get endosperm. So this is the unique feature of angiosperms. Now, this PEN further forms endosperm, which is also called as PEC. That is primary endosperm cell. This is called as cell now. Now, this is a unique feature of the angiosperms, that the two unitedness is going there. Two fertilizations are going there. One is here and second is here. That means two male gametes are there. One is uniting with the egg cell to form a zygote. And another male gamete, the second male gamete, is uniting with the polar nuclei to form a 3N structure called endosperm. As the two fertilizations are taking place, therefore it is termed to be a double fertilization. It is a unique feature in angiosperms. No other plants show this type of fertilization. In this, one thing we have to remember, very important, that will be later on, will be given you the more details about this. What happens is this, that the first step is syngamy. And the second step is endosperm formation. That means when the zygote is formed, then only the endosperm will be formed. Endosperm is used for the development of zygote. That means when zygote is formed, then only the endosperm is formed, and therefore there is no wastage in the angiosperms. Whole of the endosperms will be utilized for the development of zygote to embryo. This is very important. You know what is the fate of a zygote? We have to remember that PEN changes into PEC and PEC changes into endosperm. That means the resultant of a triple fusion will form an endosperm, and the resultant of a syngamy will form a zygote, and the zygote develops into the embryo. This is what the double fertilization is. Is that very clear to you? In the next section, we will discuss another topic. Thank you.